And a written report from the radiologist. The radiologist didn't say anything about that. So now my husband and I look at each other and it goes, well, was that nothing? So the only thing of clue is when I was talking to the doctor, he said, you know, we've come a long way with heart problems. My 35-year-old son was just discharged from the hospital today with after open heart surgery. And so, okay, so we bring Chris, so we're gonna bring Chris home and we're gonna get all these tests done again and we're gonna nail this down. He's either got problems or he doesn't. I don't want this, whatever test we have to get done, even if we have to pay for it ourselves, we're gonna get this done. Something that I've been wanting to do since he was 2 years old. And so the kid opens the door and spits. Every time he spits when we're walking to the car from the hospital. We stop at the door to get his stuff and he spits. And he won't eat or drink. He drank a little thing of yogurt smoothie, you know, that you can get in the grocery store. Goes to bed, has only taken NyQuil. The next morning, he won't even talk. And he's spitting into a cup. I finally put him to bed with a towel so it could just drool out of his mouth. And I'm thinking, his mouth is just too sore to swallow. That was my wrong step. That's the way my interpretation went. So the next morning, Steve says, my husband says, well, why don't we take him to the emergency room? I said, no, not a life or death emergency. We need to take him to urgent care first. So I took him to urgent care. The doctor looks at his mouth, turns to me and says, and I just got his mouth open far enough so I could see his right tonsil, and he was complaining of pain here, was enlarged. He said, I hate to do this to you, but we're gonna send you to the emergency room. I'm just wondering if I need to call for an ambulance. I said, what? He said, his tonsil is so big that it's in danger of blocking his airway. So I drove him to Sutter General. They called an ENT specialist. They put him on Dilaudid IV for pain. And he was dehydrated, having not eaten anything solid since Thursday or drunk anything since 2 the day before. And the ENT comes in, uses 2 different syringes and withdraws 15 mils of pus from his tonsil. He had a peritonsillar abscess. He was admitted overnight and we brought him home yesterday. He's feeling great. They stopped off at the Coonies on the way home from lunch. So after we get the tonsillar abscess diagnosis, I looked, I said, Chris, I said, what did they say when they looked at your tonsils at Napa? He goes, Mom, they never looked in my mouth at Napa. I said, they probably spent $20,000 running all of these cardio tests because that doctor was, which I don't mind him being sensitive to the heart issue, I appreciate being thorough, but never looked at the obvious. Here's a kid that's coming in having been sick with a sore throat for 10 days. I said, did you tell them that? He said, yeah.
That's annoying.
And I was just like, wow.
Not okay.
So I don't know what the insurance, I told my husband, I said, it's a good thing if you had car insurance and you keep turning in that they pop your premium up.
Yeah.
I said, I mean, it wasn't, I hope they don't do that for this because it wasn't our call to admit him, you know, over there for the cardiac workup. And we're still gonna do the cardio stuff because I don't know what he saw with the enlarged hepatic vein and all of that, but anyway,
Well,
that was my experience this weekend.
That wasn't causing him to faint, though, the whole throat thing?
No, I think it was just being sick and dehydrated.
Yeah. And then
So
the
this was
shower.
Friday, and then the hot
Yeah.
steam, you know, it was an open shower, it wasn't a steam room, but still the same event. Your skin is hot, your blood vessels dilate to release the heat. And if you're already compromised with illness and low blood pressure from
What if there's no illness involved in fainting in the shower? That doesn't seem normal to me.
It's orthostatic hypertension occurs with low blood pressure volume. So like if you've had surgery and had some blood loss, or you have low blood pressure anyway, when you stand up suddenly, that things can go gray. So if you have normal blood pressure, standing in a hot shower, you shouldn't faint. The loss of bowel control is the flood of norepinephrine. So my concern was the doctor said this happened in the car.
Who's driving?
His roommate who doesn't have a driver's license.
Fabulous.
And so he said, well, I'll just, it's 15 minutes down the hill, and Chris is like, dude, you don't have a license, I won't be with you in the car to say this is an emergency and you're taking me to the hospital. It'll be a car that doesn't belong to you and you don't have a driver's license.
You're going to jail.
He waited till his brother came by and picked him up. His roommate's brother came by and picked him up. So, but what happened, actually, it wasn't in the car. He went back to his room and he was still standing talking to his roommate that the loss of bowel control occurred, which is still, shouldn't have happened. I said, how long after you fainted? And he said, well, I don't know how long I was unconscious, but I had been in the room about 2 minutes. So whether that's still left over, I mean, that takes a flood of norepinephrine. I think I haven't gotten Justin Moore's okay on this because he's more of a physiologist than I am, but people always assume that when you go to the bathroom, it's sympathetic because people talk about wetting your pants when you're scared to death. Going to the bathroom or going either one or two is a parasympathetic response. But from what I've read is there's so much norepinephrine, it causes constriction of the blood vessels. And so the nerves then are not being able to function, like being numb, and then the muscles dilate because of relaxation. They don't have the nerves triggering them to contract and not. So sympathetic response is normally to contract, sympathetic to the muscle. So, but the response is constriction of the blood supply to those muscles, then blocks the nerves from working. So when your mouth is dry, it's because the blood vessel constriction to the salivary glands decreases the amount of fluid that can come into those glands from the blood supply. So when you do secrete saliva, it's not very much volume kind of thing. So yeah, we're still going to get the coronary, the cardiology exams done because there's just enough, we've had enough of, questionable activity and stuff that.
So it was like a cyst on his
Peritonsillar abscess. So, collection of pus and bacteria. He didn't have a fever, even when he was admitted on Friday. The neutrophils basically proliferate and form a wall that prevents the pus from spreading out and getting into the bloodstream and getting into tissues. And so that part was walled off and just collected there as this big, essentially large pimple of pus. And then the reason he couldn't open his mouth, somebody in my morning class knew the word, it starts with a T. But the reason he couldn't open his mouth was that the inflammatory process had spread out of the tonsil to the pterygoid muscles and to the tissue around. That's why it hurt back here. I thought it was referring pain to his ear, but it was all along the back of his ear where the joint is, and the muscles along the side here. Well, no, it was not in the joint itself. It was the muscles, and he still can't open his mouth very far. So in fact, when I was reading up on the procedure of aspiration yesterday, they said if they aspirate more than 3 mils, then they should go back the next day and aspirate again. And they got 15 mils, but they never went back in. So he was seen by the hospital doctor yesterday, but he hasn't been seen by ENT, so I'm a little hesitant to send him back, where he got misdiagnosed in the first place without making sure that he's on IV antibiotics and oral antibiotics. The most common bacteria that causes that is streptococcus, which is negative for, but there's also anaerobic bacteria that can get a kid in those deep crypts. You can get anaerobic bacteria from. So, yeah, it was a big learning lesson.
case study.
Yeah, there you go. He already has been one case study. All right, so for those of you coming late, I was just talking about my weekend with Chris. Heart issues and tonsil issues that fit right into what we've been talking about. So we have 2 things that we're looking at today. We're going through non-specific defense, all right? And then we're looking at T-cell, which is specific defense. And on Thursday, we'll finish up with B cells. And then we'll talk about different types of immunity with antibodies on Thursday and have a case study, and that'll be Thursday's activity. So non-specific defense refers to the body's defense mechanisms that don't require recognition of specific shapes and molecules that we call antigens. Antibodies may be involved, but it's recognition of the general class of the antibody rather than the antibody-specific recognition. So many textbooks talk about the first line of defense as epithelial tissue, and I don't want this one. Mechanical barrier. And remember, epithelial tissue has cell-to-cell junctions. So we have the blockage, the integrity of the membrane is maintained by those cell-to-cell junctions. We'll go through all of these, so you don't need to copy that down as a list right there. So on the first line of defense, we have that mechanical barrier. So we're talking about the skin, the mucous membranes, all of which have an epithelial layer. So esophagus and skin, transitional epithelium of the bladder is multiple layers. In the stomach or the small intestine or the respiratory system, we typically have tall cells, even if they are just a single layer. Now, the clarification here is that many textbooks include the chemicals on the surface of the skin as the second line of defense. All right, to me they work at the same time. So I include them as two components of the first line of defense. All right, so the first line of defense happens before the pathogen actually gets into the body. So it includes a mechanical barrier formed of the cell-to-cell junctions of epithelium. And in conjunction with that, chemicals that we find in sweat, tears, saliva, mucus secretions of the respiratory system, the pH of urine, digestive juices. So there will be, the acid, there will be enzymes, antimicrobial components found in all of these that function to even decrease the number of pathogens on the surface of the skin and decrease the likelihood that they would pass through. So I include both of these under the first line of defense. The second line of defense would be after that integrity is broken. So once a pathogen actually passes into the body, then we have to attack it before it multiplies. And so this can be cellular, or it can be non-specific. Well, it's all non-specific, but it can be fever, complement proteins. And so under the second line of defense, we have the cellular components which provide phagocytosis. And these are the neutrophil, Christopher's white blood cell count was elevated, but not tremendously. It was 12,000. So probably showing that the infection was limited to the abscess rather than, it wasn't off the wall, like 20,000 white blood cells. And his neutrophils were 84% of the total white blood cell count. So normally it's around 70 I gave you. So, and we normally see neutrophil elevation with bacteria. And that's what, and probably, I haven't gotten the lab results back from the culture, but it was probably a bacterial infection. So neutrophils, macrophages, and eosinophils are primary phagocytic cells. And that's just illustrating in phagocytosis and the breaking down of the microbe by lysosomal enzymes. Okay? Non-specific. So the macrophage is not recognizing, it doesn't have a receptor on its surface. It will only internalize certain bacteria that have a particular protein surface component. All right, in addition to these cells, we have non-specific NK cells that are not phagocytotic. Sounds like psychotic. Phagocytic. They kill these cells in the same fashion that a T killer cell does through the release of chemicals called perforin. So why isn't this a specific reaction? We have an antibody here, all right? But notice that the NK cell's receptor is recognizing the end of the antibody, not the surface of the pathogenic cell. This is still non-specific. Remember I said the end of the antibody is the constant region? And we have classes of antibodies that we'll talk about on Thursday. So immunoglobulin, IgA, IgE. So all IgEs, which are elevated in allergies, will have the same constant region. All IgGs, which will have the same constant region. All right? So the receptor of the NK cell is recognizing that constant region. It's not specific to an antigen. It's a broad recognition, so it's not considered to be specific, even though it still releases perforin like our T killer cells that we'll talk about later. So I'm going to put this up in the corner. We have 3 illustrations of antibodies being involved in a non-specific reaction. So I just want to keep a tally of those. So this is our pathogen, bacteria, whatever. It has an antigen on its surface. That antigen has been recognized and interact by an antibody. And that antibody has been recognized by the receptor of an NK cell. Okay? Which is going to release a chemical that results in rupture of the cell. But that is non-specific because the NK cell is not recognizing the antigen, it's recognizing a common protein, the end of the antibody. And I'll put the other 2 examples up there in a short time. Okay, in addition to cells, the body defends itself with fever. All right, so that's why you don't want to take a fever-reducing drug, such as Tylenol or ibuprofen, unless the temperature is above 100, sometimes 100.5, depending on what your normal temperature is. So 98.6 is standard, considered normal level. My daughter's is about 96.9, 97. So 98.6 for hers is not normal, it's elevated, all right? So if her temperature is over 99, she's running a fever. But for most people, 99, 99.5 should not be treated. If you've got kids, you should be giving them a fever-reducing agent because this is actually helping them, okay? So it decreases the bacterial activity, replication, increases the flow of blood through the body, bringing more white blood cells to the area with a higher blood flow as the heat is trying to be released from the body surface. So if it gets above 100, all right, to 101, then you want to bring it, you want to keep it down below that. I remember getting yelled at by an ER nurse when I took my daughter in. She was about 2.5 or so, and she had a temperature of 103. And I'd given her Tylenol, I'd put her in a cool tepid bath to try to reduce her temperature, and it still wasn't keeping it below that. So I took her into the ER, it was on a weekend, and this nurse was yelling at me. And I was like, I brought her here, you know? What are you yelling at me for? I did what I could in sequence, and when that didn't work, I brought her here. So, all right, and then the inflammatory response itself, which was covered in the textbook in skin. So all the healing processes we're not looking at there, but Specifically for the inflammatory process, I'm interested in the mast cell and basophil release of histamine. And histamine's role is to increase, well, two things that work together. It increases the blood flow to an area, as well as increasing the permeability of the blood vessels, okay? So this is the redness and swelling that you often see in an inflammatory response. And by vessel, even though it says capillary there, remember we know it's the post-capillary venule. So yesterday at Sac State, one of our models tipped over, and it was a homemade, it was a skeleton to which yellow wires had been attached to simulate the nerves, kind of like our wiremen. And it's a special project by one of the students during the summer, and the yellow, they were insulated wires, had been attached to the skeleton with those little plastic ties like they use for handcuffs and, you know, things, the locks, not the twisty things, but the locks. And yeah, and they had sharp edges, and this model started to fall over, and my student caught it, and it scraped her arm and made a shallow cut. And instantly, her entire wrist turned red and started to itch. Well, I don't know what was in that plastic that she had that instant contact dermatitis. I told her to, you know, she'd had basophil, not basophil, she had had Benadryl before, and you know, she could take an antihistamine, but if it got worse, to go to the health center. But that instant redness was due to histamine release, all right, in response to mast cells and causing dilation and blood increased area and then increased permeability. Really wasn't swollen yet. All right, that provides the white blood cells will leave through diapedesis, and then there's a chemotaxis, so they're going to migrate up the concentration gradient to get to the area. And then we're back to our cellular effect. So again, all of this is non-specific response. No receptor antigen recognition required. Here are some of the chemicals that are involved. So prostaglandins can be released just from the blood vessels. And dilation refers to L and A there. Plasma bradykinin is found in the blood and released by endothelial cells. Histamine we already talked about. Heparin can be released by the endothelial cells or produced by the liver. And so again, there's nothing here that's actually killing the pathogenic bacteria or viruses, but increasing the availability of the white blood cells to that area. All right, then we have proteins, most produced by the liver, that are present in the plasma. Well, the first example actually isn't produced by the liver, but produced by cells primarily that are infected by bacteria. They can be released by macrophages too, but primarily I want you to associate them with virally infected cells. This was the panacea that was going to cure cancer back in the 70s when interferon was found. Because it doesn't prevent, it doesn't kill the cell that contains the virus, but if we look at this sequence over here, here's a body cell infected by the virus. It reproduces within the host cell by causing the DNA to reproduce its own proteins, and those are reassembled and the viruses hatch from that cell and then are free to invade and infect other cells and cause them to, and we have this snowball effect. The cell that's infected at the same time that it's producing new viral proteins also produces various types of molecules known as interferon molecules. Like I said, they don't protect the host cell, but but they will be released and bind to new nearby cells that, again, don't block the virus from coming in, but block that new host cell from making new viral proteins. So it basically stops that rapid increase in the number of viruses. And that allows the cell to get a handle on the viral infection before it takes over the entire body and makes you sick. So it's thought, well, we just have to give, there's certain cancers like leukemia primarily that's caused by viruses, and we could use interferon and we've cured leukemia. Well, obviously that isn't the case, but interferon is used as part of the treatment plan, and similar drugs such as acyclovir that help to limit viral production can be produced. So we don't have any, we can't treat viral infections with antibiotics, as you know, because those kill bacteria. So if you have a viral infection, you can't take a drug that kills the virus. All we can do is try to limit its ability to replicate. Well, we can't kill off a cold virus. When one of my sons was an infant, he would get these fevers. Every few weeks he would get a fever of 101 to 103. And after 4 or 5 of them, I take him to the doctor. They say, oh, it's a virus. You know, take him home and make sure he has fluids and let it run its course. So my mother-in-law was visiting one time and Chris had a fever. She said, we've got to take him to the doctor. And I said, okay, fine. Come with me, we'll go to the doctor, but the doctor's not going to do anything but say, take him home and the virus will run its course. And sure enough, that's what happened. Because we don't have drugs that kill viruses, but this can help prevent the high number of viruses infecting other cells. Okay? So interferon is cell-produced, but in the blood we have another type of series of proteins known as complement. So if you've had microbiology, you've probably gone into it in more detail than this. But complement is very similar. Whoops, I didn't want to do that. Oh well, I'll redo it. Complement is very similar in that it's a cascade, like our coagulation sequence. So one molecule gets activated, that activates another molecule, and so on. And we call that an example of positive feedback. All right, so this cascade just gets bigger and bigger and bigger. Also, like blood coagulation, there are 2 ways of turning on this cascade. Do you have a question?
Yeah, sorry, it's about interference.
Uh-huh.
Are they actually like protein?
Uh-huh. Yeah, so they're a gene, the infection by the virus will cause a gene to be activated, copied by messenger RNA, and the cell will make these interferons that it normally wouldn't be making. So, with coagulation, we have the intrinsic and extrinsic pathway. So the intrinsic was just the contact of the blood with a foreign surface like glass or the collagen fibers. And we had the extrinsic pathway, which required the addition of thromboplastin or tissue thromboplastin to trigger the clotting factor activation. So complement activation also has 2 pathways, all right? These are known as the classical and the alternate pathway. And we're going to spend most of our time on the classical. Not very difficult. Remember I talked about the antibody recognition over here as a nonspecific use of antibodies. Here's another nonspecific use of antibodies. Okay? In the classical pathway, we're going to use this for bacteria. Let's say that a particular type of bacteria has invaded the body. It's been recognized by B lymphocytes through a process we'll talk about on Thursday. And the B lymphocyte has made antibodies. Okay? A year, 6 months later down the road, same bacterial type infects the body. It gets coated by these antibodies. Notice the constant region is what's sticking out. And so this constant region comes in contact with a complement protein known as C3. This is the classical pathway. I'm showing this event right here. So when C3 comes in contact with the antibodies, the Fc fragment of the antibody, it splits into two components. It's that contact with the Fc that causes that fragment to split, and that's the classical pathway. It's nonspecific because the Fc, the constant fragment, covers a whole variety of different antibodies. Okay? Again, as we'll talk about on Thursday, IgG antibodies all have the same constant region. IgA antibodies all have the same constant region, and so on. So the specific region of the antibody, this would be a specific interaction. The antibody binding to a molecule on the surface of the bacteria. That's specific. This is nonspecific, okay? Even though it involves antibodies. This was nonspecific here, even though it involved antibodies, because it involved the constant region of the antibody, which is not specific to that particular antibody. So when C3 is activated, it splits. Another similarity to coagulation, the numbers are in order. Okay, so they're, again, the order in which they were found. So it forms a small molecule known as C3b, and another molecule formed as C3a. C3b itself can coat bacteria. So here's our bacterium. And the C3b is going to stick to it like this. That process of coating, whether it's with the antibodies or whether it's with the C3b, is known as opsonization. Okay? So both of these are opsonization. Opsonization never killed anything. What it does, it makes it easier for phagocytic cells to recognize it and phagocytose it. So the coating with the antibodies, the coating with the C3b, doesn't directly kill the bacteria, but it increases the likelihood that it's going to be internalized by the macrophage or by the neutrophil. And that's what kills it, when the enzymes in those cells break it down. So opsonization is one of 3 ways in which complement can protect the body. Okay? So that was one. C3b interacts with complement C5, and it splits C5 into 2 smaller pieces. C3a and C3 and C5a together will interact with mast cells. And mast cells will, as you know, release histamine. And we have the inflammatory process. So that's the third mechanism or result of having complement activation. It's just an increased blood flow, more neutrophils, more macrophages coming to the area. But histamine is not going to kill cells either. They're not going to kill the bacteria. It's just triggering a pathway by which those can be killed. Okay? Finally, complement C5b plus C6, now we're finally getting something in order, C7, C8, and C9, form a donut. This donut is known as a membrane attack complex. Or MAC, another acronym for you to know. And microbiologists humorously refer to this as a MAC attack. It's not the cell being hungry for a hamburger. That gets inserted into the plasma membrane of the bacterial wall and causes rupture of the cell. So this results in, this is the only one that directly kills the cell of the 3 different pathways. Now, having gone through all that, just like those factors, you don't have to know these numbers of complement and the order in which they work. Okay? I want you to know the 3 events that they trigger. So I want you to know that the activation of complement will trigger opsonization, the inflammatory process, and direct cell rupture via the donut or the MAC attack. But you don't have to know which specific complement does what. Okay? So the same thing happens with the alternate pathway, except in the alternate pathway, the bacterium and its coat, the little sugars on its coat, react directly with C3. There is no antibody component to that step. All right. So like the coagulation, the extrinsic pathway, some other factor had to be involved, our tissue thromboplastin. In the classical pathway for complement, that other factor is the antibody. In this pathway, the antibody is not required. Okay? And in your body, both of them are working. Okay? It's just going to occur faster with the antibody. Or the effect is going to be faster. The complement is more likely to be activated coming in contact with the antibody than it is with the bacterial wall by itself. So on your lecture notes, it says specific. If you would write it, cross out the word specific because I don't want you to be confused. It's actually a non-specific component. But under classical pathway where it says specific, cross that out and just put requires antibodies. Because it really is still non-specific. It's interacting with the constant region.
That's, you said for classical.
Classical, it says specific in parentheses. Cross out the word specific and just replace it with antibody triggered. Or antibody, something to fill you in on that.
Classical is faster.
Uh-huh.
And classical is not specific.
They're both non-specific in the sense that even though classical requires antibodies, it doesn't require recognition of the antigen component to trigger the complement.
How would the alternative C3 interact
With the coat, so the polysaccharides, the sugars that are already part of the bacterial coat. So in the classical pathway, the antibody might recognize that, or it might recognize some other molecule on the surface of the bacteria. You have a question?
So that's not, alternate is not specific because it's not.
Neither of them are specific.
Because for alternate, it's not recognizing the antigen, but
Just the fact that there's a sugar there. It's not a specific sugar shape that it has to interact with for any bacterium. So if that was specific, it would only recognize a specific sugar shape. Any other sugar would not work. Okay? All right? And then these are both generalized. These are opsonization or inflammation itself kills the bacterium, whereas with the MAC attack, that directly will result in the rupture of the cell. So here's just a view of that MAC attack. So it allows fluid to rush into the cell and rupture it and burst it. Okay, so before we take a break, let's start the T cell component by talking about a term we've been using over and over and over. So I am on page 64 of your lecture notes. Can I erase the board? All right, so we've been talking about antigens ever since we started talking about blood typing. And antigens are a component of specific resistance because they are recognized by their shape from the receptors of either our T cells or our B cells. Okay? So if I draw a cell, and let's say I draw a bacterium, okay? And on the surface of this bacterium is an antigen. Okay? Now, the definition of antigen is rather circular reasoning. An antigen is a substance which stimulates the immune response. Well, what's the immune response? The activity of a T cell in response to an antigen stimulation. I mean, it doesn't tell you anything. So, here's our bacterium. Here's our antigen. And now let's say that we have a activated T cell. And after break, we'll talk about how T cells become activated. It means it's capable of responding to the antigen. It has to be primed, if you will, to do that. T cells and B cells have receptors on their surface, and it's totally random. It's based on the genes that are expressed inside that cell. And all of their receptors are going to be exactly the same. And they are pre-programmed to recognize a specific antigen. If it doesn't have that shape, then it doesn't interact with that antigen. Okay? Now notice that I didn't have it interact with the entire antigen. Antigens have different shapes on their surfaces. All right, if I were to draw that same antigen as closely as I can over here, a different cell might recognize, have a receptor for that surface. Same antigen, but this cell is recognizing a different component of it. That subset of shapes of a particular antigen is known as an epitope. So, pregnancy tests have gotten more specific. Less false positives and false negatives, okay? And this is done through the use of what are known as monoclonal antibodies. And I'm only saying this to illustrate what we mean by, so the HCG hormone, human chorionic gonadotropin hormone, is our antigen. And if we want to raise antibodies to it to use it in ELISA, we would take that molecule and inject it into a mouse. They're often used for this purpose. And along with some chemicals that increase the sensitivity of the mouse's immune system. And then the easiest way to gain access to these lymphocytes making the antibodies is to kill the mouse and take the spleen out. And it's going to be large with all of the white pulp involved in this response. Mash it, like with a potato masher. And then not dissolve, but dilute those cells in growth medium. So if let's say we have 1,000 cells per mil, all right, of growth medium. So if we have a tiny little 1,000 cells, 1,000 lymphocytes in here, and I dilute that into a liter of growth medium, now I'm going to have a ratio of 1 cell, okay, right here, a ratio of 1 cell to 1 mil. Actually, what's done is microliters are taken because that would waste a lot of growth media. And all of these cells, not all of them, but a lot of them are going to have marker receptors that match this antigen. But some might match that epitope, some might match that epitope. And it's more likely that there would be cross recognition between another antigen that had a similar surface. And so I want to limit this to a cell that only recognizes that particular epitope right there. So I take this solution and I have several wells, and I take 1 mil and I put 1 mil in each well. Now my ratio is about 1 cell per mil. So on most of these wells, I'm going to have 1 lymphocyte. Some might have 2, some might have none. But most of them will have 1. And remember, each cell, all of its receptors recognize the same epitope. So some of them may not even recognize this particular bacteria. Others will recognize different antigens, but they're all going to have just the same receptor, the same type of response. These cells will multiply, and mitosis is just a form of cloning. So when this cell multiplies to 500 cells, they're all identical, and they're all making the same antibody, all right? So when I harvest these, I have monoclonal antibodies. If I have a solution that has antibodies to this region and that region and that region, they're all to the same antigen, but it would be polyclonal. It would require different lymphocytes to make those antibodies, okay? So that gives us more specificity. So that's really the only reason why we're concerned about that, but I just wanted to illustrate the difference between an antigen and an epitope. And all of our T cells and our B cells already come with these receptors. So if you see TCR, that means T cell receptor. BCR is a B cell receptor, all right? And it's already on that cell before it ever meets whatever that receptor matches to. That's totally random, okay? All right, I'm not so worried about antigenic determinants. You don't have to really worry about that term, just the epitopes. So these are the cells that are involved in the T component of our immune system. The T helper cell, the T killer cell, and the T suppressor cell. In this diagram here, this is what we're going to be talking about today and Thursday. All right, I'm going to turn all the lights off just to make it a little, all right, I don't know how to do this anymore there. So remember we've used the phrase APC. APC stands for antigen presenting cell.